Change Fellow, and she's been here
3
3
weeks.
weeks already. So this is going to be a lot of her work from her postdoc as well as her graduate school studies. So Mansa got both her bachelor's in botany and her master's in forestry in India. Then she got her PhD in Yale in the Donahue lab. And then she did a postdoc at St. John's University in New York with Daniela Hosworth, switching from more ecology to kind of understanding how organs are fused at the molecular level. So here she's going to do her postdoc, kind of fusing all those things that she's done before and bringing new things into it using our collections outside. But today, honestly, she's going to talk mostly about what she's already done and a little bit about what she might be planning to do. So welcome, Mansa.
Can people hear me? Okay. And can people see my pointer? Yeah. Okay, great. So the distribution of organisms on Earth is not random. For example, Rafflesia, the biggest flower on the Earth, is only found in Southeast Asia. And then penguins are only found in the southern hemisphere. So why are organisms found where they are found, or why are they not found where they are not found? Well, the distribution of organisms is a complex interplay of several factors. It depends on the intrinsic properties of the species, such as its physiological traits, its anatomical traits, its genomic makeup. It depends on the geological history of Earth. It depends on its climatic history. The distribution depends on the interaction of species with other species. And of course, there's always an element of chance. So my interest lies in teasing about these factors to understand what determines basically where organisms are found at both large and also at smaller spatio-temporal scales. And I have been doing this mostly using a group of organisms called honeysuckles, which are scientifically known as Lonicera. And I'm sure many people in the room know what honeysuckles are. They are known for their colorful, fragrant flowers with nectar. And I would say that even people who don't know what honeysuckles are have probably smelled them at some point of time. And that's because so many honeysuckle species are, so many sweet-smelling honeysuckle species are highly invasive throughout the world, including here. So there are about 140 species of Lonicera, which are mainly distributed in the northern hemisphere, but then they are also and they are mostly found in the temperate forests, but then they also occupy some other biomes. So geographically, Lonicera is found in all the tertiary relict areas in the northern hemisphere. Tertiary relict areas are all the regions where plant communities from the tertiary period have survived. So, which means plants from about 66 million years to about 2 million years ago have survived. So Lonicera is found in all those areas. It's found in Western North America, it's found in Eastern North America, it's found in Southwest Europe, East Asia. And besides that, it's also found in some other area. So Lonicera is found in this circumboreal region, which I've marked in yellow. It's found in these Mediterranean chaparral type climates, both in North America and also in Europe and North Africa. There are some honeysuckle species in Mexico, and then there are many honeysuckle species in Central Asia. And majority of the honeysuckle species are found in the temperate forests, but besides that, they also occupy some higher elevation biomes of dry alpines and wet alpines. There are a few honeysuckle species in chaparrals, some in taiga, and there are also a few of them which live in these swampy, boggy areas. So we wanted to understand in which part of its wide distribution did honeysuckles originate in, and then how did they come to occupy its current the current geographic and biome distribution. And in order to answer that question, we needed a phylogeny, but the largest existing phylogeny had sampled less than 50% of the species, and then many nodes were not resolved in that tree. So we first built a bigger tree by sampling about 80% species using a technique called rat sequencing. And in rat sequencing, we basically take the DNA and we chop it up into small pieces using these enzymes called restriction enzymes. So then we make tree from that. So this is showing that tree. It's mostly well supported, and I'm gonna quickly talk about this phylogeny and how it relates to the taxonomy of the group. So the last worldwide taxonomic treatment of Lonicera was done more than a century ago by Rader, actually here at the Arnold Arboretum, and at that time he was growing many honeysuckle species from throughout the world for his work. And he had divided Lonicera into two subgenera, one called Periclaminum, which is the small one, and another called Chemiseraces, which contained all the other honeysuckle species. And the basis used by him for making these two subgenera was that Periclaminum species have flowers in groups of 3 subtended by fused leaves, and Chemiseraces species have flowers in pairs in the axils of free leaves. So we found both these subgenera to be monophyletic. And Rader had divided this big subgenus, the Chemiseraces, into 4 sections, of which we found 3 to be monophyletic. So you can see that the one in purple is monophyletic, the one in blue is, the one in green is, and the one in orange is clearly not because it's showing up in 2 parts of the tree. And then Rader had divided all these subgenera and sections into 23 subsections, of which we sampled 22, and we found that only 5 are monophyletic. And then we dated this tree. And from that, we inferred that honeysuckles originated about 40 million years ago. And we found that the crown age of this larger clade, Chemiseraces, is actually more than twice as old as the crown age of the smaller clade. So this big clade evolved about 37 million years ago, whereas the small clade evolved just about 15 million years ago. And then we used this tree to understand how Lonicera came to occupy its current geographic distribution by doing historical biogeography. And for that, we divided the distribution of Lonicera into 6 areas: Western North America, Eastern North America, Mexico, Europe, Central Asia, and then East Asia. And all these numbers in the bubbles denote how many species are found in each area and how many of those we were able to sample. And then this is showing the result of the historical biogeography analysis. The colors here are the same as the colors here, and to make things simpler, warmer colors denote parts of Asia and cooler colors denote Europe and North America. So you can see that this big clade mostly has warm colors, very less of cool colors, meaning that this clade mostly occupies parts of Asia, and there are very few species here in Europe and North America. On the other hand, this small clade has very little of warm color, so it has very few species in Asia, and most of its species are found living in Europe and North America. And we inferred that Lonicera most likely evolved in East Asia, which is this yellow color. So essentially, we found that the 2 main clades of Lonicera followed very different evolutionary paths, both in terms of both spatially as well as temporally. Okay, so now pay attention to all these stars after the species name, all these asterisks. So these denote all the species which are found in North America or New World. So we can see that this big clade entered New World 4 times, but then none of these entries radiated. Whereas the small clade entered New World just once and spawned at least 13 species. So all in all, we found 5 Old World, New World disjunctions in Lonicera. Out of these 5 times, 4 times the movement seemed to have happened from the Old World to the New World, and in the 5th case, we are not sure about the directionality. Now, all these movements between the Old World and the New World and the other way around could have taken via the 2 land bridges which used to connect them, the North Atlantic land bridge and the Bering land bridge. And judging by the ages of when all these movements happened in Lonicera, it seems like it more likely used the Bering land bridge. But all these movements could have also happened via long distance dispersal, especially because Lonicera species produce these strikingly colored juicy berries which birds seem to really love, as you can see here. Okay. Now I want to show you an interesting case of intercontinental movement in this part of the Earth. So there's a species called Lonicera involucrata, which is found in Western North America, and we found that the sister species is actually on the other side of the Pacific Ocean, endemic to these small islands here called Sakhalin Islands, and this species is called Lonicera tolmetchevai. So we we looked at photos and specimens of these two species, and we found that actually morphologically very, very similar. And they also seem to be occupying a similar kind of habitat. So Lonicera involucrata for a big part of its range is found living in these very acidic waterlogged soils. And Lonicera tolmetchivai is also found to be also reported to be living in waterlogged soils in areas flooded by a river that flows here called Time River. So all this put together provides a compelling case of intercontinental movement. And interestingly, the timing of this of splitting of these species closely matches that of the severance of this Bering Land Bridge. We traced evolution of certain traits which could have facilitated migration into new areas. So we looked at fruit color, whether the ovaries are free or they are fused, whether the species change their flower color or not, and if the species are shrubs or they are climbers, and if they are deciduous or evergreen, and also we looked at ploidy. For the sake of time, I'm going to talk only about one of these traits, which is flower color change. So about 30% Lonicera species change their flower color. Most of them change it from white to yellow, but then there are some species which change it from white to red, some from yellow to orange, some from white to purple and other colors. And we traced evolution of that, and this is showing that. So yellow means no flower color change, orange means flower color change. So we found that this trait, the evolution of flower color change, evolved at least a dozen times, but intriguingly, none of the species in the New World changed their flower color. All the species which change their flower color are found only in the Old World. And then we asked how Lonicera came to occupy its current biome distribution. And again, we did ancestral state reconstruction. Yellow denotes temperate forests, and other colors denote other biomes and biome combinations. So you can see that this tree is mostly yellow. So temperate forest biome is the dominant biome, and it was also found to be the ancestral biome for Lonicera. So what we found essentially biome-wise is that there are three main clades. This one clade moves to chaparrals, and this clade, the one in green, stays within the temperate forest. And then this big clade moves into all the other biomes where Lonicera is found. So it's found in dry alpines, wet alpines, taiga, and also swamps. Okay, now I'm going to pivot to another part of the talk, where I'll speak about Lonicera biogeography at a smaller spatial and temporal scale, and also at a smaller phylogenetic scale. And for that, I'll take you to Asia, to these mountain systems which I'm sure everyone knows about, the Himalayan Hingduan mountain systems. So these mountains started forming about 50 to 55 million years ago when the Indian plate went and rammed into the Eurasian plate. And this massive collision created some of the highest mountain peaks on Earth, such as Mount Everest. And so not surprisingly, many high altitude plant lineages are found living in these mountains, and many of them are endemic to this region. But it's very hard to study their taxonomy and therefore any aspect of their evolution, because these plants are very hard to collect. And there are two main reasons for that. The first is that these plants grow in very remote peaks and valleys, which just take a lot of resources and time to reach and collect them. And then this is a trans-boundary region, meaning that these mountains cut across several countries. So they cut across India, Nepal, Bhutan, Myanmar in the south, China in the east, there's Tibet, Xinjiang, Kazakhstan, Kyrgyzstan, Tajikistan, Afghanistan, Pakistan, and north and west. But then plants don't care about political boundaries, so most of the species are found in at least more than one of these countries. And each country has its own permit system. It takes from 1 month to more than 6 months. Depending on the country. And then resources like herbarium specimens and floras are not easily available online. And therefore, studies looking at the entire region are very few, and that hinders evolutionary studies in this region. So to fill a small gap, to solve a small part of this problem, we decided to figure out how many species make up a group of honeysuckles called radial flowered honeysuckles, or the clade Isoxylostium. And these honeysuckles are endemic to the Himalayan Hingduan mountains. And as their name suggests, these honeysuckle species have perfectly radially symmetrical flowers as opposed to all the other honeysuckle species. And this is where this group lies in the phylogeny. It's a very small clade, as you can see here. So this is a very small group, and all species are united by having either pink or either pink or white radial flowers, but then they vary in other traits. The, the, the length of the stigma and stamen varies a lot. Some species have blue fruit, some have red, some have orange, and then species can have small leaves, big leaves, and then leaves. Some species have two opposite leaves, some have leaves in whorls of 3. And then they occupy different kinds of environments. So dry temperate forests, wet temperate forests, dry alpines, wet alpines, etc. So how many species make up radial honeysuckles? So the taxonomy of the group is messy. There are at least 5 species complexes, which I've marked here in different colors. 2 of these complexes are very complex. They show a lot of morphological and ecological variation, and they have about 3 varieties each. Some scientists think that the 3 varieties of the red species are actually 3 species. And this is how they are all distributed. So different colors denote different species and different varieties. And I'm going to keep using the colors to talk about them, so you don't need to register their names. So you can see that the radial flowered honeysuckles are found throughout the Himalaya, which goes from about here to about here. And then they're found in the Hengduan mountains, which is somewhere here. And then there's also one species which goes up to Central Asia. So we collected multiple populations of all species and varieties, except for one species for which we could collect only one population. And all the collected populations are marked by these black rings. So you can see that we were able to collect from throughout the range of these species. I, along with my collaborators, collected in the Himalaya, which is in this part. And then for the Hengduan and the Central Asian populations, we were able to get them from herbaria, some botanical gardens, and from flora of Hengduan mountain project. So I collected in the Himalaya, and it's incredibly huge area, and it's stunning. And I'm going to show you photos of some places where I collected the plants from, which will give you some idea of the habitat diversity of this group. So this is a cloud forest near Mount Everest in central Himalaya. This is a glacial lake in the eastern Himalaya. This species is one of the radial flowered honeysuckle species. This is a very wet temperate forest in central Himalaya. This is a valley in western Himalaya. It's one of those valleys where Himalaya goes from being wet to dry. And then this is the extreme west of western Himalaya, and it hardly receives any rainfall, as you can imagine. But there's a species of radial flowered honeysuckle which grows here. And I'm going to give you some idea of the field collection. So some populations were easy to collect, but most of them were not. For example, to collect just a single population in eastern Himalaya, it took 16 days because it took 7 days to reach the location, 7 days of hiking, and 2 days being stuck in landslide, which is very common. In this trip, we stayed in caves, and at one point of time, I fell down from a cliff and almost died, but then got stuck in a bush. So almost every year of my PhD, I went to collect for at least 3 months. So in all, I spent about an year collecting all these plants. And that is me, happy after spotting the population. Cannot say the same about the plant. Okay, so now I'm going to show you the result of all the collection. So we collected about 109 populations in total, and we did rat sequencing of them, and this is showing a maximum likelihood phylogeny. And I told you that taxonomically there are 5 species complexes, but if you see here, there are 4 colors. There's blue in 2 places, there's yellow, there's red, and there's white, there's green, and there is no white. So what's going on? First of all, why is blue showing up in 2 places? So we found that actually one of the varieties of this blue species complex is not part of it at all. It's actually more closely related to this yellow species. And this was not expected by us, because see how these are distributed. So this light blue variety was thought to be part of this dark blue one, which just lies next to. But then genetically, we found that this light blue variety is actually more closely related to the yellow one, which is on the other side of the Tibetan plateau. So we try to understand what kind of environments these species live in, and we found that both the yellow and this light blue variety occupy these drier Tibetan plateau grassland kind of environments, whereas this dark blue species is found in actually wetter, lower elevation environment. So even though this light blue one is right next to the dark blue one, environmentally, climatically, it occupies similar areas as the yellow one. Going back to the tree, we found that the other two varieties of this this blue species complex were mixed with each other. As you can see from the two different colors, there's this purple and the blue one which are mixed. And then when it comes to this red species complex, we again found that the three varieties were mixed with each other. So which you can see from the colors, there's red and there's pink and there's this third variety with this yellowish color. And then surprisingly, we found that the white species was embedded in this red species, but this is based on only one population. So future studies should actually collect more populations and verify this. We also did structure and PC of SNPs, and that gave us the same result, which is that the same 5 species were found clustering with each other. We also did BPP and FST, which are some of the ways to figure out the limits of the species, and they also seem to support these 5 species. And I want to show you something interesting. So we sampled a population from here, which we thought belonged to the yellow species, but then in structure and PC of SNPs, it was actually found to be clustering with the light blue species. This could signal past admixture, given that the ranges of the yellow and the light blue species do not overlap today, at least based on the herbarium data. We tried to figure out how these species could have been distributed in the past at the last glacial maxima. So this is showing the distribution of the yellow species for last glacial maxima. This is showing it for the light blue species. And it seems like they could have overlapped in this area. So we would actually have to go and collect more samples from there to figure out exactly what's going on. But if admixture does get supported, then it would mean that these 3 species, the yellow, the light blue, and the dark blue one, are forming what is called a ring around the Tibetan plateau. And this mountain plateau system actually has the perfect conditions for formation of rings, because there's an inhospitable Tibetan plateau in the center, surrounded by all these habitable deserts and mountains. And in fact, people have found a ring species of bird in this region, a greenish warbler. So how many species make up radial flowered honeysuckles? So it was thought that there are 5 species based on how they look and what kind of environments they occupy, but after looking at the genetic data of multiple populations of most of these species and varieties, we found that they are 5 species, but not the same 5 as initially thought. And that is because this white species based on one population was found to be part of the red one. This light blue variety turned out to be a new species, and because none of the other varieties held up. So all methods confirm that there are 5 species, and I told you that this group shows a lot of morphological variation. So we traced evolution of all those traits, and we found epimorphies for all the clades within the radial flowered honeysuckles. For example, these 2 species evolved fully fused ovaries from free ovaries, and then I told you that it was surprising to find yellow and the light blue species to form a clade, and we found an epimorphy for them, which is that both of them have these small lanceolate leaves where you cannot see the secondary veins very well. And then the stigma and stamen length also support 5 species, because with the exception of 2 species, each species has its own unique combination of stigma and stamen length. So this blue species and this red species independently evolved these deeply embedded stigma and stamens. In the yellow species, both the stigma and stamen are exerted, which is also the condition for all the other 135 honeysuckle species except one species. And then this new species has stigma deep inside, but then the stamens reach the level of the opening of the flower. And in this green species, both stigma and stamen reach the level of the opening of the flower. And all these different length combinations most likely relate to the pollinators. We also found evidence of barriers and geographic movements in the radial flowered honeysuckle. So we found that clades within the species were sorting geographically. For example, we found 3 clades in this red species, one Western Himalayan, one Central Himalayan, and an Eastern Himalayan. And based on that and other species, we found evidence of biogeographic barriers within the Himalaya. So one barrier between Western Himalaya and Central Himalaya, and another between Central Himalaya and Eastern Himalaya. But why do these barriers exist? And the answer is moisture gradient. So Asian monsoon evolved about 20 to 35 million years ago, and what happens every year during monsoon is that moisture-laden winds start from the south, go towards the east, but then by the time they reach the west, they become very weak, and then there are these highest mountain peaks on the earth blocking them. So as a result of all of this, the eastern part of the Himalaya looks like this, very misty, gets a lot of rainfall, whereas the extreme west looks like this, very barren, doesn't get a lot of rainfall. And all this likely imposes all these barriers in the movement of the species. And there is also a dip in elevation beyond this point in the Himalaya. And it also affects the actual distribution of the species. For example, this yellow species occupies the innermost, driermost range of the Himalaya called the Trans-Himalaya, and then this green species is only found in the wetter parts of the Himalaya. And in fact, when we look at the amount of precipitation all these species receive, we find that the two main clades differs significantly. So this clade with the dark blue, yellow, and the light blue species receives much less precipitation as compared to this clade with the red and the green species. The clades also differ significantly with respect to the elevations they occupy. This clade is found at higher elevations as compared to this clade. And then they also differ significantly with respect to the kind of the kind of climates they occupy. So this is showing temperature seasonality. So this clade is found in much more seasonal climates as compared to this clade. And when we look at all the bioclimatic variables together, on all the 19 of them, we find that the two clades sort of separate on the PC1 axis, which is most heavily weighted by precipitation and temperature. So all these differences between the two clades with respect to the elevations they occupy, the kind of environments they live in, must have had some effect on their population sizes during the glacial periods. Higher elevation species must have undergone more glaciation and hence more extinction as compared to the lower elevation species. And we do find some signals of that when we do niche modeling for these species for past and last glacial maxima. So this is showing the current distribution for the red species from the lower elevation clade. And this is showing the distribution at last glacial maxima. And you can see that there was some contraction in the size of the populations. And when we do the same thing for this blue species from the higher elevation clade, this is distribution at present, this is distribution at LGM, we find that there was much more contraction in the population size. And we probably see signatures of these niche related contractions between the two clades in the dated tree as well. So this is the higher elevation clade occupying colder, drier, and more seasonal climates. This is the lower elevation clade occupying warmer, wetter, and less seasonal climates. And we find that the branches the branches leading to the crowns in the higher elevation clade are comparatively longer as compared to this lower elevation clade, which could probably be because of more extinctions here. And then when we look at the branches leading to the present day populations, we find that they are actually smaller here as compared to here, which could be probably because of the extinctions and then the expansion from the bottlenecks. And it's most suggestive for this population, for the species here, which is the one that occupies the extreme west of Western Himalaya. And this was probably one of the harshest places for species to survive in this entire region, not only because it underwent a lot of glaciation, but also because it was very aridified. People have found this star-like phylogeny with like very short branch lengths in some other organisms that occupy the Western Himalaya, such as the Himalayan langur. And we also detected the directionality of movements in some of these species based on the phylogeny. It seems like most of the Himalayan species move from the east towards the west, and this primarily Hengdawan species most likely evolved in the south and then moved northwards and then also moved eastwards towards the Himalaya. And I want to tell you one last thing on radial flowered honeysuckles based on this fun observation I had in the field. So there's this species which I've been showing in red, and it has the widest distribution of all the Himalayan species. It goes all the way from the west to all the way to the east, and it's mostly in temperate forests, but it also occupies higher elevation biomes at the edges of its range. And it's found in sympatry with three other species that are found in the Himalaya: the yellow one, the blue one, and the green one. And what was very fun and also frustrating in the field is that this species closely looks like the species it's found growing with. So here's the red species growing next to this green one and looking kind of like it, and they were both about 10 feet. Here is the red species again growing next to the blue one and then kind of looking like it, both were 3 feet. And this is the most dramatic of all. Here is the red species again, the same one as here, and here it's growing next to this yellow one, and it kind of looks like it. And these were both less than 20 centimeters. And when I was going through herbarium specimens, I found that this red species was frequently misidentified as one of the other radial flowered honeysuckles. And we don't know what underlies this crazy morphological and ecological variation, but it must have a role to play in its wide distribution range. Okay. So now I'm going to briefly talk about what I was doing in my last postdoc. So honeysuckles are known for their fragrant, colorful flowers. They're known for their pretty viney species, and they're known for being highly invasive. But something which not many people know about honeysuckles is that they show rampant fusion of certain floral and extra floral organs. So many species have free leaves, but in some species the leaves are fused. Some species have free ovaries, some have partially fused ovaries, some have completely fused ovaries. And then bracteoles, which are these leafy appendages that subtend the flowers, show many kinds of fusions. So bracteoles in some species are free. In some species, bracteoles of the adjacent flowers fuse. In some species, bracteoles of the same flowers will fuse at the outer margin of the flower. In some species, all 4 bracteoles, so the 2 bracteoles here and the 2 at the back, fuse and form a cup around the ovaries. In some species, this cup of bracteole expands and then completely covers the ovaries inside. So this green structure which you see here, these are not ovaries, this is actually a cup of bracteoles. And in a few species, this cup of bracteole becomes fleshy and then takes on different colors. And then all these fusions are phylogenetically clustered. So leaf fusions only happen in the small clade, and then all the bracteole and ovary fusions happen in this big clade with the exception of one species. And the question is, why does Lonicera show all these fusions? So boundary between organs across angiosperms and also outside of them has been found to be maintained by a gene called QQ. The less the expression of QQ, the less effective is the maintenance of boundary, and then more the organ fusion. So less QQ, more fusion. In angiosperms, there are 2 separate copies of QQ. One is called QQ12 and one is called QQ3. And these have overlapping functions. There are some differences. But when it comes to expression, there is this big difference that QQ12 expression is tightly controlled by microRNAs, whereas the expression of QQ3 is not. So collaborators led by Aaron and Daniela tried to figure out the evolutionary history of QQ in Lonicera and its relatives, and they did that by pulling out QQ copies from transcriptomes, whole genomes, and genome skimming data. And this is what they found. They found that the outgroup species of Lonicera have QQ1, 2, and QQ3. But then in Lonicera and its close relatives, QQ3 seems to be completely lost. And that's the copy which is actually not tightly regulated by microRNAs. They also designed baits for capturing the QQ, and that gave us something like this, the coding region and the upstream and downstream regions. So when I joined as a postdoc, I started analyzing the data, and I actually found some putative QQ3 copies from Lonicera and its immediate relatives. So this is showing QQ3 fragments from Lonicera and its close relatives. This is showing it from the outgroup. So for outgroup, we were able to mostly capture the entire QQ3. But then for Lonicera and its immediate relatives, we were only able to find like very small fragments, and there were stop codons in between exons. So it seems like QQ3 is not completely lost, but it seems to be on its way out. It seems to be pseudogenizing. And I want to give a shout out to Mark, the master's student who was working with me there. He designed this pipeline to align all these fragments. So most of the available aligners were not working very well for us because they're either designed for coding fragments or non-coding fragments, not for both of them at the same time. But Mark's pipeline was able to align this. And what we have been doing is we are looking for causes of these free and fused morphologies in Lonicera by looking at the QQ12 gene, not QQ3 because that's very small to do anything. So we are looking at enhancers. Enhancers are these structures that bind upstream of the coding region at these sites called enhancer binding sites, and they increase the rate of transcription of a gene. So enhancers will increase the rate of increase QQ expression, and that's gonna maintain the boundary, and which will give like all these free morphologies. But if there would be any kind of disruption in the enhancer binding sites, then QQ expression would be less. The boundary won't be maintained as effectively, and that would give us all these fused morphologies. So we're waiting for the entire base data to come back. Once we have that, we'll have it for Q and 24 other genes which are involved in this regulatory network and with fusions. And we'll have it for about 80% Lonicera species and about 80 outgroup taxa. And I've also collected RNA-seq data from free and fused organs from different Lonicera species, which will shed more light on all of this. So that's all I have from this work. I would like to take the last few minutes to talk about what I'll be doing here over the next 2 years. In case anyone has any suggestions on how to make the project better, please let me know. Also, if anyone has any suggestions for any pots of money I could apply to, external, internal, small, big, please let me know. So across life forms, we find that closely related lineages which have diverged from a common ancestor evolved to occupy very different kinds of biomes, many times despite having similar spatiotemporal opportunities and similar dispersal capabilities. For example, this tree is showing two clades, clade A and clade B. Both of them started out in the temperate forest. Clade B stayed within the temperate forest, but then clade A also moved into deserts. And the question is this totally random, or can we actually pinpoint specific evolutionary events in this lineage's history which will explain this uneven biome occupancy? It is possible that lineage A acquired some traits that helped it to move to the deserts, which lineage B lacks. Now, if this biome would be a harsh one, then it will make it a little easier to answer this question of uneven biome distribution. And that's because harsh biomes impose very strict filters on the lineages that can occupy them, which makes it a little easier to identify these enabling traits. For example, alpine biomes are one of the harshest biomes for plants on the earth, mostly because how cold it gets there. So alpine species across different plant families, lineages have acquired this short height in order to survive the cold, and they have thin xylem vessels so that the damage from the freezing of the water and rupture of the tubes is minimal, and they have high stomatal density in order to make up for the small leaf size, and they show expansion of gene families which are associated with common alpine stresses like cold and drought. So Lonicera is a good system to study the question of uneven biome distribution in closely related lineages with respect to alpine biomes. And that's because even though Lonicera transitioned to alpines, both to these dry ones and the wet ones, multiple times, at least 5 times independently, all those transitions are restricted to this big clade. So all the 3 clades started out in temperate forests, but then only this clade moved to alpines. And here is a more granular view of that. So this figure is showing the evolution of biome on the Lonicera phylogeny. The clades are same as the clades here. So yellow denotes temperate forest, this brown denotes chaparral Mediterranean kinds of biomes, and the blues denote the alpines. Light blue is wet alpine, dark blue is dry alpine. So we can see that there have been 5 transitions to the alpines, but only in this big clade, specifically in these 2 clades. And the question is, did this big clade evolve any kind of enabling traits that allowed it to move to alpines, specifically these 2 clades? So we can look at the similarities and differences between these clades with respect to the relevant alpine traits. So if we talk about temporal opportunities, these 2 clades, this one and this one, evolved about 30 million years ago, whereas this clade evolved just 15 million years ago. So this clade had half as less time as the other 2 to move to alpines. If we talk about spatial opportunity, by which I mean if they all have species living in temperate forests adjacent to the alpine ones, all these 3 clades have such species, but then this clade has very few such species. In terms of dispersal capabilities, all Lonicera species produce berries, but then they have, seed number varies. It can go from 1 to 5 to 8. So they might or might not have similar dispersal capabilities, and we also don't know whether there are any differences between the number of fruits these species produce. When it comes to habit, there's this big difference, which is that this big clade has shrubby species, whereas these 2 have climbing species. And then the climbing nature of these 2 clades might have hindered them from moving to alpines, because there are not many things to climb on an alpines. And then the climbing habit may also have some associated anatomical features that might have hindered their transition to alpines, but then we don't know about their anatomy. When it comes to the stem pith, these two species have hollow pith. This big clade has solid pith, but then it has this big non-alpine clade where the pith also turns hollow. And there are hollow pith species in the alpines, so having a hollow pith might not be a hindrance by itself, but then we don't know what their exact anatomy looks like, what their vessels look like. And then these two clades produce accessory buds and this clade does not. And probably having extra buds is better for moving into alpines because terminal parts are most prone to frost damage. There's a lot of damage which happens because of the cold. And then if we look at just this big clade, if we look at clades within this clade, then there are differences with respect to the size of the bud, the number of bud scales, shape. And there must be other differences between these clades. There must be some other anatomical differences, physiological, genomic ones. There must be differences with respect to the bud and the growth architecture, but they haven't been studied yet. So what I have done is I have identified certain traits that are found across alpine plants from like different lineages, which I'm going to call alpine relevant precursors, and I'm going to examine them across the alpine and the non-alpine clades throughout the Lonicera phylogeny in some specific species. I'm going to look at anatomical precursors. For that, I'm going to take the cross section of leaf and stem. I'm going to look at leaf peel, and I'm going to compare the number and size of xylem vessels, stomatal density, thickness of cuticle and palisade layers between alpine and non-alpine species. I'm going to look at genomic precursors by doing RNA-seq. I'm going to identify differentially expressed genes between the alpine and the non-alpine species. I'm going to look for any potential, any expansion or contraction of any relevant genes and gene duplications in them. And I'm going to compare the cold tolerance between the alpine and non-alpine species. And then I'm going to look at the bunch of morphological precursors using herbarium specimens and also living plants. So I'm going to look at some bud traits like the position of the bud, how many accessory buds are there, how many bud scales are there, etc. I'm going to look at some growth architecture traits like the height of the plant, what's the dimension of the leaves are thick or thin, etc. And then I'm going to use hidden Markov models to test whether these traits acted as precursors to alpines, and if so, where they arose in the phylogeny. And I'm also going to do some correlation tests between the traits and the biomes. And you're probably wondering where I'm going to get all the material from. So the great thing about the Arboretum is that it has many non-alpine species growing here, spanning the entire phylogeny of Lonicera. And all the species growing at the Arboretum are marked by these stars. And then I'm going to collect the alpine species and their closest temporal relative from the field. And I'm also going to try growing them at the Arboretum. There are alpine species growing in botanical gardens around here. And with respect to the herbarium specimens, again, luckily for me, Harvard University Herbaria has one of the biggest collections of Lonicera worldwide. And I've identified some other herbaria which also have good collection of Lonicera species. So the study will shed light on whether this uneven alpine biome distribution in Lonicera is random, or is it due to the evolution of specific traits in the history of this lineage. And if we do find alpine precursors, then it will inform us on the timing and the order in which all these precursors evolved, and hence their relative significance. And I told you that there are both the wet and the dry alpine species of Lonicera, and there are at least three sister species pairs that are found in wet and dry alpine, speaking broadly. So irrespective of the first and the second outcomes, this work will inform us what the anatomical, physiological, genomic, and morphological differences exist between the dry and the wet alpine species, and also between the wet and the dry alpine species and their closest relatives in an evolutionary context, which to my knowledge has not been done before, especially for the species growing in the Himalaya. And with that, I would like to thank the Lonicera team, which includes my PhD advisor Michael, my postdoc advisor Daniela, Wendy, my Indian collaborators Amit sir, Rawat sir, my undergrad and master student at St. John's University, Herbaria Botanical Gardens throughout the Northern Hemisphere, and the Flora of Hengduan Mountain Project, especially for sending us tissues through the thick of COVID. I want to thank Patrick, the Yale Herbarium manager, for helping with all the tissue loans, or all the specimen loans, the funding agencies, and I want to thank all of my field assistants and also some friends and family who helped me collect plants from throughout the Himalaya and made sure that I returned back alive.
Any questions?
Yes.
That was awesome. I'm really excited for you to be here and to work on these things. I'm interested in, you touched on that you have looked at this, but you didn't talk about it today. I'm interested in how polyploidy interacts with different traits in this family, and then also where you find polyploids in the phylogeny and across the range.
Yeah, I think I have a slide with the polyploidy.
And then, as like a follow-up to that, do you
Yes.
expect polyploidy to have some association with alpine habitats?
If you see this third figure, that's showing polyploidy. So most of the Lonicera species have the 2n as 18, and there have been many evolutions of tetraploids, and there are also some odd numbers. With respect, so we tried to figure out if all the species living in more harsher environments had higher ploidy levels, and we did. So we did not find very strong evidence of that, but there seem to be, but species which seem to be occupying harsh habitats seem to have higher ploidy levels. There's a species complex called Lonicera cerulea, which is circumboreal and also goes down to like Japan and Himalaya. In those species, people have found that that most of the low elevation species, the lowland species, have a lower ploidy level as compared to the ones which are at higher elevations. So, not very sure about this, but could be. Yes.
In the beginning, you were talking about color change in the flower.
Yeah.
Am I understanding that the flowers changed as they evolved, or the color of it?
So flower color changes over the course of 2 to 3 days after the flower pops up. Yeah, so not in an evolutionary context, but like, and it has been found that it's been found to honestly signal the pollinators about the quantity of the nectar. So the nectar is higher before the flower color changes, and it becomes lower as it keeps on changing. Yeah.
And do you plan on visiting any alpine areas around here, or just sticking to the mountains?
I would like to, not because there are any honeysuckle species which grow in alpines here. All the alpine honeysuckle species are found in the Himalaya, but I would love to go and explore alpine areas.
Okay. And one last question.
Yes.
What type of shrub saved your life?
So I don't know. It was it was like 9 p.m. at night, and it was very dark. And all I know is that I felt upside down, so my legs were up, and I couldn't like do anything. And I could also hear the shrub creaking under my weight. So I don't think it's a very strong shrub. That's what I'm trying to say.
Luckily it was there.
Yes.
In your phylogeny, very impressive phylogeny from your PhD, you had one Lonicera spinosa that just seemed to be kind of hanging out in the middle between the yellow and the light blue. It was very different from all the other ones, and I'm just curious if that was like found somewhere weird or.
Oh, you're talking about that one which is like in the. Okay, let me go back to that. This one, probably. Yeah. So I did not get into this, but there's a species which is reported here called Lonicera alberti, which has some traits from like Lonicera minuta. The leaves look like it, but then its stigma and stamen length matches Spinosa. So there could be a gradation here, but then we don't know much. These species are so understudied that we don't even know what the fruit color of Lonicera minuta is. Like that's how little these things are studied. But this specific specimen had actually stigma and stamen popping out like Lonicera spinosa, but it had these longer leaves like Lonicera minuta, and it also had hair inside the corolla, which doesn't happen in spinosa but happens in minuta. So it might be something in the middle. At least morphologically, it seems like that, and also genetically, it seems like that. But we would need to go and collect more populations from there. Yeah. Yes.
How much within species variation is there for the fusion phenotypes that you were talking about?
So most species have either fused or free or like partially fused, but there are some species which show a lot of fusion within the populations, and I also found within the individual that there is a lot of variation. Um, in one of the species, I actually found flowers fusing. So the two flowers would completely fuse, and there would be this big mega flower with like two stigma and like 10 or something stamen. So yeah, so there is within species variation.
That makes sense. It just seems very plausible that if your hypothesis of a related enhancer is true,
Yeah.
that this is something that could be possibly reoccurring or re-evolving within species, say like the transposome.
Yeah.